So it's a real pleasure to present to the this Keystone Symposium, and this particularly with regard to the Vin Prize, which is a truly remarkable prize, and I was honored to win it earlier on this year. So what I'm going to talk about today, and very briefly, is a journey that I've had from physics to lipid nanoparticles to gene therapy, and this has literally been gene therapy, including the Pfizer-BioNTech vaccine. It's literally been a 50-year journey. I have a number of conflicts of interest, as indicated at the bottom. Now, many people say that the mRNA vaccines are delivered in tiny bubbles of fat, and this is a little bit of a simplification. The actuality is that they are certainly very tiny. They're 100 nanometers in diameter, smaller, 100 times smaller than the typical cell, but they're really not bubbles of fats. They're rather sophisticated systems that are made of membrane lipids. And all of this started 50 years ago. I got my PhD in physics in 1972, and I really had the feeling I wanted to do something different. The most interesting problem seemed well outside the field of physics. And so I got a postdoc to go to Oxford in 1973 to study the properties of lipids found in biological membranes, really knowing very little, almost nothing about either lipids or biological membranes or anything in biology for that matter. One of the first things I did when I got to Oxford was help build an NMR machine that I then used to study properties of lipids. And why did I find membrane lipids interesting? Well, biological membranes rely on a bilayer that is composed of membrane lipids, obviously enough, that separates the inside from the outside. And without this permeability barrier, cells couldn't exist. And so membrane lipids are certainly extremely essential to life. Now, what I studied was lipid polymorphism and lipid asymmetry. Couldn't quite figure out why there were so many different, this is still true today, so many different species of lipids in membranes, and why in particular some membrane lipids were adopted non-bilayer structures. So why are those lipids there, and why do they adopt those structures? And I also got interested in lipid asymmetry, the fact that lipid composition on one side of a membrane is different than the lipid composition on the other. And I wondered if we could generate that asymmetry in response to ion gradients, and what are the consequences of these asymmetric distributions. So 50 years later, this work led to lipid nanoparticles that enable mRNA vaccines. This went through, obviously enough, a number of stages. First of all, packaging RNA, DNA into lipid nanoparticles. This is in many ways work that's still continuing on in my laboratory. The Onpattro story, which took place between in the 2000s, this was to treat a disorder known as hereditary transthyretin-induced amyloidosis, and then the COVID-19 vaccine story. So I'll very briefly just go through those events. Now, encapsulation of DNA and RNA in lipid nanoparticles requires positively charged cationic lipids. For obvious reasons, you have a highly negatively charged polymer. You need a positively charged lipid to get this whole thing encased in a lipid environment or hydrophobic environment. The big problem with this is that positively charged cationic lipids are highly toxic, and so we had a big issue, or how do we encapsulate nucleic acids in systems that are not highly toxic? Now, we developed an ionizable cationic lipid as part of our basic work on lipid asymmetry. And this lipid had the property that at low pH, say pH 4, the acidity of a lemon, then the lipid was positively charged, but at neutral pH, it was not charged. And so this is just indicated here. And what we found was that we could load nucleic acid polymers into lipid nanoparticles at pH 4, where they're positively charged. And then the contents were retained when we brought the pH up to physiological pH values. And so we could load these lipid nanoparticles with siRNA and mRNA, and as everybody is well aware, this allows us to practice many forms of gene therapy with siRNA to inhibit production of a given protein, and messenger RNA to induce production of a particular protein. And so this is quite a wide range of possibilities in terms of therapeutics. So first we used LNP containing siRNA to treat this disorder that I mentioned, transthyretin-induced amyloidosis, a very nasty disease, and it's indicated here. And it's caused by production of a mutant form of transthyretin in the liver. So in a very simple hypothesis, we say, okay, if we stop production using siRNA to inhibit production of transthyretin in the liver, we should reduce the deposition of the fibrils that are caused by the mutation, which then deposit in neural tissue and in cardiac tissue. And maybe we get some stabilization, possibly recovery from the cardiomyopathies and neuropathologies that evolve. This was a spectacularly successful clinical trial. The change in neural impairment score, or the improvement, much better than placebo by values indicated here, 10 to the minus 24, 1 over Avogadro's number. There's no doubt this drug works. And in all the secondary endpoints, this was also a spectacularly successful medicine. These results were announced in 2017. So we concluded from this that Onpattro, this lipid nanoparticle formulation of siRNA, is a stable and possibly curative therapy for a previously fatal disease, a hereditary disease. This as a result, of course, of these results, then the FDA approved this drug in 2018. So we then thought, and this was a company I co-founded called Acuitas, we thought, okay, if we can deliver lipid nanoparticles containing mRNA and have a gene silenced, containing siRNA and have a gene silenced in the liver, maybe we could package mRNA and have a gene produced in the liver. And remarkably, we found we could use the liver to produce whatever protein we wanted. And this is really quite a spectacular statement to make. But to cut a long story short, we were then approached by Drew Weissman from the University of Pennsylvania and Katie Curico from BioNTech, who needed a delivery system to enable all the work that they'd done on mRNA therapeutics as vaccines, which they produced a modified mRNA to reduce immune activation and increase gene expression. And the basic thing was that we have a delivery problem. How do we get mRNA coding for viral proteins into muscle immune cells in vivo? And this work commenced, a part of it was on influenza virus, the other on Zika virus. And these results were published in 2017, showing that the lipid nanoparticle mRNA systems could produce highly effective vaccines when the mRNA coded for the Zika virus pre-membrane and envelope glycoprotein, as is indicated up here. And so this was this really gave some considerable impetus to the possibility that lipid nanoparticles containing mRNA could be excellent, could provide excellent vaccines. And so for Acuitas, this led to a collaboration with BioNTech to develop an influenza vaccine. And then in January 2020, when the pandemic hit, then all efforts, of course, switched to develop a COVID-19 vaccine that BioNTech had already been working with Pfizer on an influenza vaccine, but of course everything switched to COVID-19, and so it ended up that the lipid nanoparticle that we developed was then used to package mRNA coding for the SARS-CoV-2, the spike glycoprotein. And as we're all aware, this was, again, a rather spectacularly successful. In November of 2020, Pfizer and BioNTech concluded a phase 3 study indicating that the vaccine was 95% effective in all age groups for preventing COVID-19 in all age groups, and ethnicities. And very well tolerated across all populations. Now, we're all well aware this has been approved in many jurisdictions now. The companies indicated they were going to produce 1.3 billion doses by the end of 2021. I think that was closer to 3 billion, and this is playing a major role. in controlling the COVID-19 pandemic. But these vaccine applications are really just the tip of the iceberg. You're going to see vaccines against many more influenza, HIV, Zika, etc. The possibilities of using these types of approaches, these gene therapy approaches, for chronic diseases, cancer, heart disease, Alzheimer's, etc., is certainly in the cards. And as we saw for transthyretin-induced amyloidosis, inherited diseases, we can certainly have the potential for doing a lot for them. And so that's really the story written in a very 30,000-foot view that of the development of lipid nanoparticles that enable gene therapies, including the mRNA vaccines. So it's really been quite an amazing journey, starting from very, very basic work on lipid polymorphism and lipid asymmetry. I have to acknowledge literally hundreds of collaborators in this effort, and particularly my very long-term collaborators, Mick Hope and Tom Madden, with whom I've worked for the last over 40 years. It's been quite a journey for all of us. So thank you very much.